So cellulose is present in plants and it is a constituent of cell wall of the plant cells. So when it is necessary, glycogen will undergo hydrolysis and produce glucose, which is used up by the body. Carbohydrates are used as storage molecules as starch in plants and glycogen in animals. So it is not present in our body or it cannot be synthesized by our body. It is supposed to be taken through the diet or food. So that is why it is called as essential. It has to be taken up. Hello everyone, this is Ambli Unnikrishnan from the Department of Chemistry. So today I am back with session 3 of the chapter Biomolecules. So in the last session we completed the discussion of glucose, right? So we discussed the cyclic structure of glucose, then we studied about fructose, its cyclic structure as well as Hayward structure. Then we moved on to disaccharides and we discussed about maltose, lactose, and we started off with polysaccharides. So we have discussed starch in the last session. So in today's session we are going to discuss about the further polysaccharides, okay, and then we'll complete the discussion of that, and then we'll discuss about the importance of carbohydrates, and we'll move on to the next important topic, that is amino acids, and the classification as well as the Zwitter Ion Form of amino acids we'll discuss in this session, right? So starting off with the next polysaccharide after starch, that is cellulose. So cellulose occurs exclusively in plants, okay? So cellulose is present in plants and it is a constituent of cell wall of the plant cells. Cellulose is a straight chain polysaccharide composed of beta-D glucose units. So all of these monosaccharides, disaccharides and polysaccharides we have studied. It is important to remember which are the monomer units of this or which are the monosaccharides of it, right? So in the case of cellulose, it is a polymer, okay, of which it is beta-D glucose units, okay, it's beta-D glucose units which are joined by glycosidic linkage between C1 of one glucose unit and C4 of another glucose unit. So this is one beta-D glucose here, this is another beta-D glucose here which is bonded between the carbon 1 and carbon 4, okay. So this is carbon 1 of one glucose molecule, this is carbon 4. So a glycosidic bond is formed between C1 and C4 of two glucose molecules, that is beta-D, and further it will continue. So a polymer of this will give you cellulose, right? So now the next one is glycogen, which is also very important. The carbohydrates are stored in animal body as glycogen. So we require carbohydrates, right? So this glucose that is essential for human body is being stored in the form of glycogen in animal body. So when it is necessary, glycogen will undergo hydrolysis and produce glucose, which is used up by the body. So carbohydrates are stored in our body in which form? It is stored as glycogen, right? It is also known as animal starch because its structure is similar to amylopectin. Amylopectin is a component of starch we have studied, right? So its structure is quite similar to amylopectin. So glycogen is also called as animal starch, but it is more highly branched. It is present in liver, muscles and brain. When the body needs glucose, enzymes break down the glycogen to glucose. So it is stored as glycogen, and when we are in need of glucose, the glycogen will break down with the help of enzymes and produce glucose. Glycogen is also found in yeast and fungi. So this you can see it's a highly branched structure. So you can see this is one molecule of glucose, and between the C1 and C6 you are having another glycosidic linkage, and here you can see between C1 and C4 another C1 and C4, and it will continue. So this quite has a similarity with amylopectin, that is why it is also called as animal starch. Clear? Now moving on to the next part, that is importance of carbohydrates. So carbohydrates are essential for life both in plants as well as animals, right? And honey has been used for a long time as an instant source of energy by Veds in Ayurvedic system of medicine. So honey was earlier used as a source of energy, instant source of energy. Now carbohydrates are used as storage molecules as starch in plants and glycogen in animals. So carbohydrates, which is very essential for human body, how it is stored? It is stored in the form of starch in plants and in the form of glycogen in animals. So when it is necessary, when the glucose is necessary, it reacts with the enzyme and forms. So it is stored in the plant body as starch and human body as glycogen. When the glucose is necessary or the energy is necessary, what happens? The enzymes will break down the starch or glycogen to form glucose, right? So cell wall of bacteria and plants is made up of cellulose, and they provide raw materials for many important industries like textiles, paper, breweries, etc. So these are few importance of carbohydrates. Now moving on to the next important biomolecule, that is amino acids. So from this name itself, what you can understand, amino and acids. So amino stands for something related to ammonia or amine group, right? And acid stands for, yes, the carboxylic acid group. So from here you get a basic idea. It's going to be a combination of an amino group and a carboxylic group. So let's understand that. Amino acid contain amino, that is NH2, and carboxylic acid group, that is COOH. Only alpha amino acids are obtained on hydrolysis of proteins. Okay, so this is a part of proteins, that is proteins undergo hydrolysis to form amino acids. That is, it means that amino acids is the ones which is combining together to form proteins. Okay, so before moving on to studying in detail about proteins, which we'll be studying in the upcoming sessions, we need to have an idea about amino acids, right? So amino acids, as discussed, it's a combination of amino group and acid group, that is carboxylic acid. So only alpha amino acids are obtained on hydrolysis of proteins. So what is that you understand from this sentence? The amino acid groups will combine together to form proteins. So before studying about proteins, it is necessary to understand about amino acids first, and then in the upcoming sessions we'll study about proteins, right? So as said, amino acids is a combination of amino group and the carboxylic acid group. So amino acids are usually colorless crystalline solids. Fine. These are water soluble, high melting solids and behave like salts rather than simple amines or carboxylic acid. So they behave as salts because they are having a base, that is amino as well as carboxylic acid, right? So there is a base as well as an acid. So it behaves as a salt rather than behaving as a base or acid. This behavior is due to the presence of both acidic and basic groups in the same molecule. So COOH, which is carboxylic, is acidic and amino group is basic. So because of the presence of both, it acts as a salt rather than acting as a acid or base. Amino acids are generally represented by a 3-letter symbol. Sometimes 1-letter symbol is also used. And the structure of some commonly occur amino acids along with their 3-letter and 1-letter symbols we are going to discuss now. So this is a basic structure of amino acid which is shown here. You can see the NH2 group, that is amino. Right, and you have a CH and this alkyl group will be different. So based on the difference of this R, you will have different amino acid groups, and here you have the carboxylic acid group. I hope it is clear. So generally you can write it as NH2 CHR and you have COOH. So this R can change. Clear? So this can be an alkyl group, it can be an aryl group, whichever is necessary. So let's discuss few examples. So the first one is glycine, okay, and the characteristic feature of side chain, that means which is the R group we are taking, based on that we'll get different amino acids. So glycine, if we are taking in this particular general formula, it is going to be NH2 CH and you have this R which is also H, right? So it becomes CH2COOH. So NH2CH2COOH is called as glycine and the 3-letter symbol is GLY and the 1-letter code is G. So either of these two you can use. Now the next one is alanine, wherein the R is CH3, methyl group. So it is represented as ALA or A. So according to the general formula, this is going to be NH2 and you already have a CH here and you will have a CH3. Yes, instead of this R, you are going to have the CH3 and you have COOH. This is alanine. The next one is valine, in which the R group, this R group is going to be CH3 twice CH. So it is represented as VAL or V. Right. Moving on to the next one, leucine, which is important, CH3 twice CH, CH2. Right. So totally 4 carbon atoms are there. That is a butyl group. Right. So it is represented as LEU or L. Now moving on to next one, isoleucine, that is an isobutyl group, that is CH3, CH, CH3, CH you have. Okay. It is represented as ILE or I. Next, arginine, it is CH3, CH2, NH2 and CH3 and CH2 thrice. So this, whatever you are going to see, this will be substituted in this R. So that is called as arginine, and it is represented as ARG or R. Moving on to the next one, lysine. This is also very important. You have NH2, CH2 4 times in the place of this R, right? So it is represented as LYS or K, right? Moving on to next one, glutamic acid. So some acid will be also there, right? So it is NH2CO CH2. This is an amide which is present, and then you have GLU and represented as E. So next one is aspartic acid. You have COH CH2 CH2, which is the alkyl group, and it's represented as ASP or D, right? Next one is glutamine. This is COOH CH2, and it's represented as GLN or Q. Moving on to the next set, that is asparagine. The alkyl group here is NH2COCH2CH2, represented as ASN or N, right? And next you have threonine, which is CH3CHOH, and it's represented as THR or T, 3-letter and 1-letter word. Next you have serine, which is OHCH2, represented as SER or S. Next you have cysteine, which is SHCH2. So you have S here, sulfide is present here, and it is CYS or C, right? Moving on to next one, methionine. It is CH3SCH2CH2, represented as MET or M. Next one is phenylalanine. So phenylalanine means you have a phenyl group as well. So it is C6H5CH2. That means you have NH2, you have CH, and instead of alkyl group, you are going to have C6H5CH2 here, and you will have COOH. So this is your phenylalanine structure, and it's represented as PHE or F. And the last one, tyrosine, and it is represented as OHC6H5CH2, that is THR or Y. Okay, so in here the important ones are mentioned here, that is valine, leucine, isoleucine, lysine, alanine, glycine. All of these are important, right? So moving on to the next set, that is consisting of ring structures, right? So the first one is tryptophan, which is very important. So this is the alkyl group which is going to be present in place of R, right? And then it is represented as TRP or W. Next you have histidine, and it's represented as HIS or H. And the last one is proline. So this is the R group which is going to be substituted. It is represented as PRO or P. So these are the examples of different amino acids which has a combination of amino as well as carboxylic acid group. So the R group that is shown in this structure, the R will be represented or substituted by these groups that we have just discussed now, right? Now we are going to see the classification of amino acids. So this can be classified on basis of two things. First one is based on their chemical nature, that is whether they are going to be acidic, basic or neutral. So first one is neutral amino acid, then you have basic amino acids and acidic amino acid. The next one is based on the dietary requirements, that is whether we have to take it through our diet, that is through the food we have supposed to take it, or it is already present in our body, that is essential amino acids and non-essential amino acids. So let us discuss about it in detail. So the first classification is based on their chemical nature. So the first one is neutral amino acid. So it contains equal number of amino and carboxyl groups. For example, glycine. So glycine is an example here. So what was glycine? The R group is H there. So you have NH2, CH2 and COOH. This is glycine. So from here you can see that there is only one amino group and one carboxylic group. So this is the number of amino group and carboxylic group is equal. That means it is neither going to be basic or acidic. It's going to be neutral, right? Now acidic amino acid. Now you can tell what will be acidic amino acid. The COOH or carboxylic group will be more, right? So contains more carboxylic groups than amino groups. Example is aspartic acid, because it is already containing carboxyl group apart from the carboxyl group which is already present in the amino acid. Clear? So COOH will be more than NH2, because of which it's going to be acidic. Next one is basic amino acid, contains more number of amino rather than carboxyl group, right? Example is lysine. So what you have to remember, the general structure is NH2CHR COOH. So, if both of these groups NH2 and COOH is equal, then it's going to be neutral. If carboxylic groups are more, then it's going to be acidic. If amino groups are more, it's going to be basic. That's it. Clear? Now, the next set of classification is based on their dietary requirements. So, the first one is essential amino acids. Essential means that it has to be taken up through the diet, right? So, they are required by the human body, but they cannot be synthesized in the body. They must be taken through food, okay? So, it is not present in our body, or it cannot be synthesized by our body. It is supposed to be taken through the diet or food. So, that is why it is called as essential. It has to be taken up, right? Example, valine, leucine, etc. Now, non-essential, of course, it will be already synthesized in our body, right? Also required by the human body, but they can be synthesized in the body itself. Example, glycine, alanine. Okay, so these are already synthesized in our body, so need not take it up through food. They are called as non-essential because it's already present in our body. So, essential ones are supposed to be taken through diet, right? So, moving on to the Zwitter Ion form of amino acids. Now, in aqueous solution, the carboxyl group can lose a proton, right? So, in aqueous solution means when this amino acids is reacting with water, or if it is present in water, what happens? Dissolution can take place, right? So, what is exactly happening there is carboxyl group can lose a proton. Carboxyl group is COOH. So, in the aqueous medium, what can happen? The proton can be lost from the carboxyl group, and we have an amino group also, well, right? So, the amino group can accept a proton. Amino group is basic. It has the tendency to accept proton. Carboxylic group is acidic. So, it has a tendency to lose proton, right? Giving rise to a dipolar ion known as Zwitter ion. So, this is very important. This is neutral but contains both positive and negative charge, okay? So, it contains both negative and positive charge, but it is neutral. Zwitter ion is neutral. So, let us understand. In Zwitter ion form, amino acids shows amphoteric behavior because they react both with acids and bases. So, let us understand here RCH CO, NH2, OH you are having. So, what is this? You can see here. This is the amino group and this is the carboxyl group you are having. Right. So, the carboxyl group has the tendency to lose this proton. So, H plus will be lost and the COO will become COO minus. And amino group is having the tendency to accept this proton. So, what is going to be happening? This H plus will be accepted near NH2 and this will become NH3 plus. Right, one proton is accepted. So, that is what you can see here. RCH will remain as it is. This will become COO minus because one H plus is lost, right? So, this will become COO minus and this proton which is lost from the carboxyl group will be accepted by the amino group because it is a base. And this is the Zwitter ion form because you have both positive as well as negative charge in the same molecule. Because of that, it is going to be neutral, but this is called as a Zwitter ion form. And it has, once it is in the solution phase, it is forming the Zwitter ion and it is having amphoteric nature in this state because it can act as a base as well as an acid because of the positive as well as negative charge. I hope this Zwitter ion form is clear for you. So, the proton will be lost from the carboxyl group which will be accepted by the amino group. So, this is the Zwitter ion form of amino acid. So, I hope that is clear. Moving on to the next part, that is peptide linkage. So, in amino acids you have the linkage which is called as peptide linkage. In carbohydrates or in glucose we have studied the linkage is called as glycosidic linkage, right? In amino acid it is called as peptide linkage. So, the amide linkage, that is CONH, formed between the carboxylic group of one molecule of an amino acid and the amino group of another molecule of amino acid by the elimination of water molecule is called as a peptide linkage, right? So, a dipeptide is made up of two amino acids linked by one peptide linkage. So, let us understand this. You have one molecule of glycine here and one molecule of alanine here. So, you can see NH2, CH2, COOH and you have NH2, CH, CH3, COOH. So, what is going to happen here is, this is one molecule of an amino acid. This is another molecule of amino acid. So, how this bond is formed is by the removal of water. So, you can see here what is going to be happening is this H2 and O will be removed. H2O is removed as water. Now, what is left behind here is going to join and there is a peptide linkage which is going to be formed. So you have CO and you have this N and H. So this CO-NH bond is called as the peptide linkage. So you can see NH2CH2 remains as it is. Water will be removed. What is left behind? CO-NH. So a CO-NH bond is formed. This is called as the peptide bond, and it is a dipeptide bond because it is formed between two amino acids and only one peptide bond will be formed, and whatever is left behind CH3 COH will be there, right? A tripeptide contains 3 amino acids linked by 2 peptide linkages. Similarly, when 4 or 5 or 6 amino acids are linked, it is known as tetrapeptide, pentapeptide or hexapeptide. Okay, so dipeptide bond means it does not indicate 2 peptide bonds are there. It indicates that there are 2 amino acids but only 1 peptide bond, right? Now, if it is a tripeptide bond means there are 3 amino acids involved, wherein there will be 2 molecules of water removal and forms 2 peptide bonds. Clear? Now, when the number of such amino acids is more than 10, it is called as polypeptides. Clear? So, let's take an example of 2-glycine itself. You have NH2, CH2, COOH, and you have one more molecule of NH2, CH2, COOH. So what is going to be happening here? H2O will be removed and a CONH bond will be there. So you have O here and H2 here will be removed. So you'll have CONH which is left behind, right? So whatever else is there, water will be removed and you will have NH2, CH2 which is already present, and you will have a CONH bond which is a new one formed between this, and you will have CH2 and COOH forming here. Clear? So the CONH bond which is formed by the removal of water between 2 different or 2 same amino acids is called as the peptide linkage. Clear? So when it's a dipeptide, there will be involvement of 2 amino acids and 1 peptide linkage will be there. Clear? So in the next session, we are going to discuss in detail about proteins. Okay, it is made up of amino acids. So I hope what we have discussed regarding amino acids is clear for you. So with this information, we are going to study about proteins. So I hope the concepts that I've covered in this session is clear for you. So that's all for today. Thank you.